Dear students, welcome to our program. Today we're going to discuss the topic antibodies, in which we'll have a look at historical perspective of antibody structure, function and properties of antibodies, different classes, subclasses and biological activities of antibodies, and finally transport of immunoglobulin A. First of all, let's discuss antibodies. Antibodies are the antigen-binding proteins present on the B cell membrane and secreted by plasma cells. Membrane-bound antibody confers antigenic specificity on B cells. Antigen-specific proliferation of B cell clones is elicited by the interaction of membrane antibody with antigen. Secreted antibodies circulate in the blood, where they serve as the effectors of humoral immunity by searching out and neutralizing antigens or marking them for elimination. All antibodies share structural features, bind to antigen and participate in a limited number of effective functions. Now let's discuss the basic structure of antibodies. Antibody molecules have a common structure of 4 peptide chains. This structure consists of 2 identical light chains or L chains, polypeptides of about 25,000 molecular weight, and 2 identical heavy chains or H chains, larger polypeptides of molecular weight 50,000 or more. Like the antibody molecules they constitute, HLN chains are also called immunoglobulins. Each light chain is bound to a heavy chain by a disulfide bond and by such non-covalent interactions as salt linkages, hydrogen bonds and hydrophobic bonds to form a heterodimer. The first 110 or so amino acids of the amino terminal region of a light or heavy chain varies greatly among antibodies of different specificity. These segments of highly variable sequence are called V regions, VL in light chains and VH in heavy. All of the differences in specificity displayed by different antibodies can be traced to differences in the amino acid sequences of V regions. In fact, most of the differences among antibodies fall within areas of the V regions called complementarity determining regions or CDRs. And it is these CDRs on both light and heavy chains that constitute the antigen binding site of the antibody molecule. Dear students, now let's discuss the historical perspective of the antibodies. The first evidence that antibodies were contained in particular serum protein fractions came from a classical experiment by A. Tiselius and E. A. Cabot in 1939. They immunized rabbits with the protein oval albumin, the albumin of egg whites, and then divided the immunized rabbit serum into 2 aliquots. Electrophoresis of one serum aliquot revealed 4 peaks corresponding to albumin and the alpha, beta and gamma globulins. The other serum aliquot was reacted with ovalbumin and the precipitate that formed was removed. The remaining serum proteins which did not react with the antigen were then electrophoresed. In a key experiment, brief digestion of IgG with the enzyme papain produced 3 fragments, 2 of which were identical fragments and a third was quite different. The 2 identical fragments, each with molecular weight of 45,000, had antigen binding activity and were called as FAB fragments. The other fragment, molecular weight of 50,000, had no antigen binding activity at all. Because it was found to crystallize during cold storage, it was called FC fragment, fragment crystallizable. Digestion with pepsin, a different proteolytic enzyme, also demonstrated that the antigen binding properties of an antibody can be separated from the rest of the molecule. Pepsin digestion generated a single 1 lakh molecular weight fragment composed of 2 FAB-like fragments designated the FAB2 fragment, which binds antigen. The FC fragment was not recovered from pepsin digestion because it had been digested into multiple fragments. A key observation in deducing the multi-chain structure of IgG was made when the molecule was subjected to mercaptan reduction and alkylation, a chemical treatment that irreversibly cleaves disulfide bonds. Initial attempts to determine the amino acid sequence of the heavy and light chains of antibody were hindered because insufficient amounts of homogeneous protein were available. Although the basic structure and chemical properties of different antibodies are similar, their antigen binding specificities and therefore their exact amino acid sequence are very different. The population of antibodies in the serum gamma globulin fraction consisted of a heterogeneous spectrum of antibodies. Sequencing analysis finally became feasible with the discovery of multiple myeloma, a cancer of antibody-producing plasma cells. The antibodies from a normal plasma cell are indistinguishable from the antibodies of cancerous plasma cell, which is called myeloma protein to denote its source. In a patient afflicted with multiple myeloma, myeloma protein can account for 95% of the serum immunoglobulins. In most patients, the myeloma cells also secrete excessive amounts of light chains. These excess light chains were first discovered in the urine of myeloma patients and were named Bence-Jones proteins for their discoverer. When the amino acid sequences of several Bence-Jones protein light chains from the different individuals were compared, a striking pattern emerged. The amino terminal half of the chain, consisting of 100 to 110 amino acids, was found to vary among different Bence-Jones proteins. This region was called variable or V region. The carboxyl terminal half of the molecule, called the constant region, had two basic amino acid sequences. This led to the recognition that there were two light chain types, kappa and lambda. Dear students, now let's discuss immunoglobulin fine structure. The amino acid sequences of immunoglobulin heavy and light chains contain several homologous units of about 110 amino acid residues. Within each unit, termed as domain, an interchain disulfide bond forms a loop of about 60 amino acids. Light chains contain one variable domain VL and one constant domain CL. Heavy chains contain one variable domain VH and either 3 or 4 constant domains CH1, CH2, CH3 and CH4 depending on the antibody class. Immunoglobulin domains are folded into a characteristic compact structure called the immunoglobulin fold. This structure consists of a sandwich of two beta-pleated sheets, each containing antiparallel beta strands of amino acids, which are connected by loops of various lengths. The beta strands within a sheet are stabilized by hydrogen bonds that connect the NH groups in one strand with carbonyl groups of an adjacent strand. The beta strands are characterized by alternating hydrophobic and hydrophilic amino acids whose side chains are arranged perpendicular to the plane of the sheet. The hydrophobic amino acids are oriented toward the interior of the sandwich and the hydrophilic amino acids face outwards. The two beta sheets within an immunoglobulin fold are stabilized by the hydrophobic interactions between them and by the conserved disulfide bond. Maximum variation are seen in those portions of the sequence that correspond to the loops that join the beta strands. These regions were originally called hypervariable regions in recognition of their high variability. Hypervariable regions form the antigen binding site of the antibody molecule. Because the antigen binding site is complementary to the structure of the epitope, these areas are now more widely called complementarity determining regions or CDRs. The 3 heavy chain and 3 light chain CDR regions are located on the loops that connect the beta strands of the VH and VL domains. The remainder of the VL and VH domains exhibit far less variation. These stretches are called framework regions or FRs. Dear students, now let's discuss antibody classes and their biological activity, starting with the first, immunoglobulin G or IgG. IgG, the most abundant class in serum, constitutes about 80% of the total serum immunoglobulin. The IgG molecule consists of 2 gamma heavy chains and 2 kappa or 2 lambda light chains. There are 4 human IgG subclasses, distinguished by differences in the gamma chain sequences and numbered according to their decreasing average serum concentrations: IgG1, IgG2, IgG3 and IgG4. The structural characteristics that distinguish these subclasses from one another are the size of the hinge region and the number and position of the interchain disulfide bonds between the heavy chains. The subtle amino acid differences between the subclasses of IgG affect the biological activity of the molecule. IgG1, IgG3 and IgG4 readily cross the placenta and play an important role in protecting the developing fetus. IgG3 is the most effective complement activator, followed by IgG1. IgG2 is less efficient and IgG4 is not able to activate complement at all. IgG1 and IgG3 bind with high affinity to Fc receptors on phagocytic cells and thus mediate opsonization. IgG4 has an intermediate affinity for Fc receptors and IgG2 has an extremely low affinity. Now let's discuss immunoglobulin M or IgM. IgM accounts for 5 to 10% of the total serum immunoglobulin with an average serum concentration of 1.5 mg per ml. Monomeric IgM with a molecular weight of 180,000 is expressed as membrane-bound antibody on B cells. IgM is secreted by plasma cells as a pentamer in which 5 monomer units are held together by disulfide bonds that link their carboxyl terminal heavy chain domains C-mu4 and their C-mu3 domains. The 5 monomer subunits are arranged with their Fc regions in the center of the pentamer and the 10 antigen binding sites on the periphery of the molecule. Each pentamer contains an additional Fc-linked polypeptide called the J chain or joining chain, which is disulfide bonded to the carboxyl terminal of cysteine residue of 2 of the 10 mu chains. The J chain appears to be required for polymerization of the monomers to form pentameric IgM. It is added just before the secretion of the pentamer. IgM is the first immunoglobulin class produced in a primary response to an antigen, and it is also the first immunoglobulin to be synthesized by the neonate. Now let's discuss immunoglobulin A or IgA. Although IgA constitutes only 10 to 15% of the total immunoglobulin in serum, it is the predominant immunoglobulin class in external secretions such as breast milk, saliva, tears, and mucus of the bronchial, genitourinary, and digestive tracts. In serum, IgA exists primarily as a monomer, but polymeric forms like dimers, trimers, and some tetramers are sometimes seen, all containing a J chain polypeptide. The IgA of external secretions called secretory IgA consists of a dimer or tetramer, a J chain polypeptide, and a polypeptide chain called secretory component. The secretory component is derived from the receptor that is responsible for transporting polymeric IgA across cell membranes. The J chain polypeptide in IgA is identical to that found in the pentameric IgM and serves a similar function in facilitating the polymerization of both serum IgA and secretory IgA. Dear students, now let's discuss transport of this IgA. The daily production of secretory IgA is greater than that of any other immunoglobulin class. IgA secreting plasma cells are concentrated along mucous membrane and surfaces. Every day, a human secretes from 5 grams to 15 grams of secretory IgA into the mucus secretions. The plasma cells that produce IgA preferentially migrate to subepithelial tissue where the secreted IgA binds tightly to a receptor for polymeric immunoglobulin molecules. This poly-Ig receptor is expressed on the basolateral surface of most mucosal epithelia, for example, the lining of digestive, respiratory and genital tracts, and on glandular epithelia in the mammary, salivary and lacrimal glands. After polymeric IgA binds to the poly-Ig receptor, the receptor IgA complex is transported across the epithelial barrier to the lumen. Transport of the receptor IgA complex involves receptor-mediated endocytosis into coated pits and directed transport of the vesicle across the epithelial cell to the luminal membrane, where the vesicle fuses with the plasma membrane. The poly-Ig receptor is then cleaved enzymatically from the membrane and becomes the secretory component which is bound to and released together with the polymeric IgA into the mucous secretions. The secretory component masks sites susceptible to protease cleavage in the hinge region of the secretory IgA, allowing the polymeric molecule to exist longer in the protease-rich mucosal environment than would be possible otherwise. Secretory IgA serves as an important effector function at mucous membrane surfaces, which are the main entry sites for most pathogenic organisms. Because it's polymeric, secretory IgA can cross-link large antigens with multiple epitopes. Binding of secretory IgA to bacterial and viral surface antigens prevents attachment of the pathogens to the mucosal cells, thus inhibiting viral infection and bacterial colonization. Now let's move on to the immunoglobulin E or IgE. IgE antibodies mediate the immediate hypersensitivity reactions that are responsible for the symptoms of hay fever, asthma, hives, and anaphylactic shock. The presence of a serum component responsible for allergic reactions was first demonstrated in 1921 by K. Prosnitz and H. Kostner, who injected serum from an allergic person intradermally into a non-allergic individual. When the appropriate antigen was later injected at the same site, a wheal and a flare reaction analogues to hives developed there. This reaction, called the PK reaction, named for its originators, was the basis for the first biological assay for IgE activity. IgE binds to Fc receptors on the membranes of blood basophils and tissue mast cells. Cross-linkage of receptor-bound IgE molecules by the antigen and allergen induces basophils and mast cells to translocate their granules to the plasma membrane and release their contents to the extracellular environment, a process known as de-granulation. As a result, a variety of pharmacologically active mediators is released and gives rise to allergic manifestations. Localized mast cell degranulation induced by IgE also may release mediators that facilitate a buildup of various cells necessary for antiparasitic defense. Lastly, let's discuss immunoglobulin D or IgD. IgD was first discovered when a patient developed a multiple myeloma whose myeloma protein failed to react with the anti-isotype antisera against the then known isotypes IgA, IgM and IgG. When rabbits were immunized with this myeloma protein, the resulting antisera were used to identify the same class of antibody at low levels in normal human serum. The new class, called IgD, has a serum concentration of 30 microgram per milliliter and constitutes about 0.2% of the total immunoglobulin in serum. IgD, together with the IgM, is a major membrane-bound immunoglobulin expressed by mature B cells, and its role in the physiology of B cells is under investigation. No biological effector function has been identified for IgD. Well, dear students, I really hope you would have understood today's lecture. That's all for today. Thank you.